When you think about your daily usage of fossil fuels, what comes to mind first? Is it your car ride in the morning? Sure, but what about something like your Gore-Tex jacket or your fish's plexiglass aquarium? We often don't realize it, but a myriad of products in our homes and lives are made using petrochemicals. This model is unsustainable, and we must find alternative ways to produce the chemicals we need. Now, one of the most promising feedstocks for renewable chemical production is all around us, wood. What makes wood a really great feedstock is that any greenhouse gases that are emitted during the production process can be offset by photosynthesis during the tree's lifetime. Biorefineries, or refineries that transform wood into energy and commodity products, do exist right now, but they're still largely unprofitable and unsustainable. That's largely due to the fact that 1/3 of wood's biomass is made up of this very large and complex molecule called lignin, which current industrial processes are unable to transform into anything useful. Consequently, lignin is treated as waste and burned. But what if we could find ways to valorize this abundant, untapped resource? This is where microbes and my research come into the picture. So interestingly, in nature, some bacteria have evolved the ability to catabolize, or in other words, eat, many types of lignin-derived molecules. In our lab, we study one such bacteria called Rhodococcus joshi RHA1, or just RHA1. We're interested in understanding how we can harness the metabolic abilities of RHA1 through genetic engineering. So what if instead of using these lignin molecules for growth, RHA1 could actually convert them into value-added chemicals? Envision a living cell factory where the blueprints take the form of DNA and the machinery takes the form of proteins and enzymes. So the overall goal of my master's project is to engineer RHA1 to transform acetovinilone, which is an underutilized lignin-derived molecule, into value-added chemicals. Naturally, RHA1 is not able to catabolize acetovinilone. However, some of its cousin species are. Unfortunately, those organisms are much harder to work with and to engineer than RHA1, but their genetic information can still be borrowed. So right now in the lab, I am taking genes from these acetovinilone catabolizing organisms and inserting them into RHA1's genome, effectively giving the cell the blueprints to an acetovinilone degrading machine. Downstream of this, I'll then be looking at other sets of genes that will enable RHA1 to transform the product of this degradation machine into valuable products. Specifically, I'm interested in engineering a cell factory to produce mucinate, which is a molecule that has broad applicability in the manufacture of plastics, fibers, and pharmaceuticals. Overall, with this work, our hope is to engineer living cell factories to convert currently wasted lignin into value-added chemicals. In this way, we can start to move away from fossil fuels and towards renewable modes of production. Thank you.